Hello everyone. We are live now, so we can start in a couple of minutes, I guess.
Okay. Good morning, everybody. I know we are going through a very difficult times, and I hope that definitely the situation will improve in a couple of weeks' time. So, as you know, that we are organizing this Science Setu event today. This is the first seminar that we are organizing today. First of all, on behalf of National Institute of Biomedical Genomics, I welcome you all here for this virtual seminar. And I sincerely expect that when the situation improves, all of you can actually visit NIBMG, which is in Kalyani, West Bengal. So we have a fantastic museum here, Genome Hall. You can all come here and visit this. But definitely when the situation improves, we expect you to visit here. So let me introduce myself. I'm Moulinath Acharya. I'm working as a faculty member in the National Institute of Biomedical Genomics. Today, let me introduce the speaker first, and then the speaker will take over. Today's speaker for the Science Setu seminar is Dr. Priyadarshi Basu. Dr. Priyadarshi Basu actually completed his PhD from the Shah Institute of Nuclear Physics in Calcutta on neurodegenerative trinucleotide repeat disorders in Indian ethnic populations, and then he moved on for his postdoctoral studies. First in the Department of Human Genetics in Virginia Commonwealth University, and then he was associate research scientist in health sciences and research in Philip Morris, US. And then he actually came back and joined the National Institute of Biomedical Genomics in 2011, where he started his research as a independent investigator. So his primary research area is broad research area, includes complex disorders, gene environmental interaction in complex disorders or common metabolic disorders. Primarily, he is interested in genetic and epigenetic causes of metabolic diseases, such as non-alcoholic fatty liver disease and drug-induced liver injury. And he is basically here, he is trying to identify early molecular biomarkers of such diseases. So without much delay, without further delay, I would like to, over to Dr. Priyadarshi Basu for his presentation. Thank you. So just a small announcement that you can actually post your questions in the chat box, and then after the talk is over, Dr. Priyadarshi Basu will look at those questions and he will answer those. Okay, thank you.
So my slide is visible now. Thank you, Dr. Acharya. My slide is visible now?
Yes, it is.
All right. Okay. So good morning, everyone. And thank you all for joining this first webinar on Science Setu. I know these are very trying times, and so thank you all. Please be careful. Please take care of yourself and your near and dear ones. And with that, I'll start my talk. So my talk today will be on the evolving field of genomics, as the title suggests. So my name is Priyadarshi, and I'm a faculty here at National Institute of Biomedical Genomics. So what I will do today is basically take you on for a journey, starting from the how the field of genomics evolved, starting from what we understood as genetics and how it has evolved to the field of genomics, and what the Human Genome Project has actually made us understand. There are lots of different things that we have started understanding since the Human Genome Project, and finally from 2013 onwards, what has been going on in the field of genomics, and how we are basically trying to translate our knowledge of genomics into personalized medicine for all. So with that, what is a genome? So a genome is the entirety of an organism's hereditary information. So you know, our hereditary information, whether it's in the form of DNA or RNA, is transmitted from one cell to the daughter cells, and also when we pass on our genome to our offspring, so to the next generation. So that entire information, starting with all those genes and all the regions that are basically do not code for proteins, everything is actually the genome, and it is encoded either in DNA. Mostly for most organisms, it's the DNA. But in certain cases, in case of certain viruses, it's also in the RNA. For example, the COVID-19 virus or the influenza virus, these are RNA virus. So instead of DNA, their hereditary material is RNA, ribonucleic acid. So the genome actually consists both of the genes, and the non-coding sequences of the DNA. So to start with, I'd like to take you through the foundational milestones in the field of genetics and genomics. Starting with this gentleman, Gregor Johann Mendel. So this, you must have all read about the pea plant experiments of Mendel. So this was the first time he demonstrated that there is something which is heritable, which can be passed on from one generation to the other. So that was Mendel's contribution, which was several contributions in the field of genetics that I'm going to talk about. So starting with Mendel. Next, but what Mendel didn't understand was that what was the genetic material. He didn't know about genes, he didn't know about DNA or RNA. So he called these factors. And he was lucky enough that he chose characteristics of a pea plant which were encoded by only one gene. So that's why he could demonstrate very beautifully how it's passed on from one generation to the other in a certain way. It was Mischer who actually identified the nucleic acid. So the thing that we know as the deoxyribonucleic acid or ribonucleic acid, like DNA or RNA, Mischer was the one who in 1871 identified those molecules. But people even didn't know that those were actually the hereditary molecules in the cells. So it took a long time after that. People used to think that it was actually proteins which carry hereditary information from one generation to the other. But it was Avery MacLeod and McCarthy's classic experiments where they demonstrated that it was actually DNA of a bacteria which can transform. So that was the first time we understood that DNA or RNA is the hereditary material. But still, we didn't understand how it was passed on from one generation to the other till in 1953, when Watson and Crick and Rosalind Franklin they demonstrated how DNA the DNA in eukaryotes is actually double-stranded, and they are passed on in a particular fashion. So if we consider in the last 120 years of what was the most important discovery of the century in the field of genetics, that would be the experiments, the demonstration of double-stranded DNA by Watson and Crick and Rosalind Franklin. So this was actually the first page of that particular paper. So now we know that DNA is made up of 4 letters. So G, A, T, and C: guanine, adenine, thymine, and cytosine. So everything in the DNA is actually combinations of these ATs and GCs. And in the human genome, in humans, the size of that sequence of ATs and GCs is 3.2 billion base pairs, which means that in each cell of our body we have 6.4 billion bases. So 6.4 billion of these letters of A, T, G, and C. So this is what the human, if you consider the human genome as a book, so it consists of 23 chapters. It's actually 46 chapters. You can consider them as chromosomes. So there are 23 pairs. 20 chapters of A and B, so there are 46 chromosomes, and each chapter contains 48 to 250 million letters. So that's approximately the size of the different chromosomes, from 48 to 250 million bases, and these are without spaces. So there are about 6.4 billion letters in all in each cell in diploid cells in the humans, and at least one copy of the book is contained in most cells of our body. The exception, of course, is mature red blood cells, which do not have any nucleus and hence do not have any chromosomes, do not have DNA at all. So to get you through a brief history, the actual term genomics is thought to have been coined by Dr. Tom Roderick. He was a geneticist at Jackson Laboratory, which is in Bar Harbor, USA. And this was in a meeting held in Maryland, where they actually started thinking of mapping the human genome, sequencing the human genome and mapping the human genome in 1986. but sequences of whole genomes had already started by then. In fact, in 1972, Walter Fires and his team, they determined the complete nucleotide sequence of a bacteriophage. Bacteriophage MS2, this is a type of virus actually. So this is in fact an RNA virus, and this was the first time that RNA virus sequence was completely sequenced. The first DNA-based genome was sequenced in 1977 by Frederick Sanger. This was the virus called bacteriophage 5X174, and we still use this particular technology, which is called the Sanger sequencing technology. Except it's not as cumbersome as it used to be. It's digital now. But that's one of the ways of sequencing which is still in practice. The first free living organism, so viruses outside the body is considered non-living. So the first free living organism that was sequenced was that of Haemophilus influenza in 1995. So from 77 to 1995, that was a long time. And since then, it was like a breakthrough. After that, the genomes have been sequenced at a very rapid pace. In fact, the complete sequences are available for around 4,000 viruses, 1,200 archaea and bacteria, and 3,500 eukaryotes, out of which 250 are mammals. And for example, the COVID-19 virus, we have sequenced close to 6,000 of strains. So that's one virus, but there are many strains which have been sequenced in our institute at NIBMG. The Human Genome Project, which was a project to actually sequence the human genome and map the human genome, that started in October of 1990. And this has been compared to the Lewis and Clark expedition, which happened in the 18th century. So these two gentlemen were mapping the Mississippi River at that time. So the Human Genome Project has been likened to the Lewis and Clark expedition of the 21st century. So in 1990, the project began. The first draft was published in 2001. And in 2003, the Human Genome Project was officially at its end because the entire genome had been sequenced by then. So there were two things. So it was an international effort, of course. There were 8 countries which were involved. And they were determining the sequence of base pairs which make up the DNA. And one of the seminal things about after the sequencing of the Human Genome Project was that before that, like during the days when I was doing my PhD, we thought that the total number of human genes was something to the order of 30,000 to 50,000. But after the completion of the Human Genome Project, we understood that the number of genes were actually much less. It was found to be about 18,000 to 23,000. The figure is current figure is close to 22,000. But then one of these genes can make many different forms of proteins, which are called isoforms. So that's why even having 22,000 genes, that can produce hundreds and thousands of different kinds of proteins. The proteins are the ones which do the cellular function in our body. It's not the genes themselves, but the genes are the ones which code for these proteins which do cellular function inside the body. So timelines of the genome sequence, I already talked to you about the viruses which were sequenced in 1972 and 77. The first plant genome was sequenced in 2000. And in 2003, the human genome was sequenced. But I would like to point out is this thing is that the human genome is only 3.2 GB, but there are other organisms in both the animal and plant kingdom which have huge size of genomes. So it's the human genome is actually tiny compared to them, and this is because they have a lot of these what we call the repetitive sequences. So these are huge chunks of repeats, which apparently is just there doing nothing. So that's why. So the human genome is it's the most complex, of course, but it's still not the biggest. So in 2011, when the first draft of the human genome was published, this is what Adam Rutherford said, that the mistake we often make is saying that the Human Genome Project was an end. In fact, the Human Genome Project was a pregnancy. 10 years later, we now have a clue what we don't yet know. The Human Genome Project might be finished, but understanding our genome is only just the beginning. And that is what I'm going to talk to you about in the next slides. So how we came to understand the human genome. So why are we studying all this? It's to cure diseases, right? So this is the realization of the genomic medicine. So I mean, for example, there are drugs which work beautifully for Caucasians but don't work very well for us. For example, some of those diabetic drugs. So what we need is specialized drugs which will work in our population. So that is what the genome medicine has promised to us. So I'm going to take you from the time that the Human Genome Project was completed and how we are going to use this to realize the realization of genomic medicine. This is called many things like bench to bedside or helix to health. So, but they mean the same thing: realization of genomic medicine. So that's the promise: healthcare tailored to the individual based on their genomic information. So to take you through that path, so how do we manage to do that? So what happened after the completion of the Human Genome Project? So first thing what we needed to do was to understand the function of the human genome sequence. So from 1990 to 2000, this was the time when the human genome was mapped. So basically, we had to understand where its sequence is coming from. You cannot sequence the entire thing in one go. So you have to sequence fragments. And after sequencing the fragments, you have to know where these fragments have gone. Which fragments comes after which and which fragment comes after that. So this actual sequencing was from 1998 to 2000. 2003. And from 2003 onwards, after the completion of the Human Genome Project, we are trying to interpret the human genome. So after the completion of the Human Genome Project, this is what scientists were looking at. So this is only 3,000 bases, which is 0.0001% of the human genome. Remember that the human genome is 3.2 billion bases. So this is just 3,000 of them. So you are looking at a huge bunch of numbers, not knowing what it really means. So that was the first task, to understand what this sequence of letters really mean. So the first thing that we at that time, what we knew was that if these are also called codons. So these are triplets of 3 letters. If they come side by side, they code for certain kinds of amino acids. And amino acids are the building blocks of proteins. For example, TTT or TTC, if they are side by side, these 3 letters, they will code for a particular amino acid called phenylalanine. So by that way, if we found a bunch of these letters which were kind of making sense because they were going to code for proteins, we know those are the regions that actually can code for something. It might not code for anything, but then that's at least one of the clues that it might code for something if these triplets of letters are all side by side together. So then we can start coloring the genome. So these are the sequences among these whole bunch that actually codes for something, codes for a protein or part of a protein, because these are also called exons. So these are parts of the gene which code for proteins. Then inside the gene also there are regions called introns which do not code for anything but are there. And if you have a bunch of these coding sequences which are coming together, which are close by, then there's a it's a fair guess that this actually means that there's a gene there. So that's how the number of genes came down to 22,000, which we now know is probably more or less the total number of genes in the human body. So what about the rest? Why is it required? Does it do anything at all? So because this coding region is actually 2% of the human genome. What about the rest of the 98%? So and why is it required? What is required for us to survive? So this is the gentleman who actually gave us a clue. He's probably the single most important individual in the field of biology and genetics, and this is of course Charles Darwin. So and this gentleman is still giving us clues to understand why the genome has evolved in the way it has evolved. So this is what Darwin said, that it is not the strongest of the species that survives, not the most intelligent that survives. It is the one that is the most adaptable to change. So if the strongest or biggest of the species were to survive, then our world would be, we would still have dinosaurs in our world. And there are many species which have existed and then gone extinct because they could not survive. The ones that have survived in this current age is the ones that have adapted to change, to the changing world, the best. So this gave rise to the field of comparative genomics. So that is why after the Human Genome Project, to understand what is important, there were a whole bunch of other organisms whose genomes were sequenced, and this gave rise to the field of comparative genomics. So we were comparing what is common to all these organisms, because they are still the surviving organisms on this earth. So there must be something common to them that has led them to survive till now. And to do that, the project which was launched was Genome 10K project. So the idea was to sequence 10,000 vertebrate species, approximately one for every vertebrate genus, and to capture the diversity of, not only to capture the diversity of the vertebrate species, but also to understand what was common among all these species, so that we understand what is important in that genome. So what this project found was that 5% of the human genome sequence is constrained across mammals or conserved across mammals. And so this 5% is extremely important for survival of all these species. So that's actually 150 million base pairs, and out of which about 1.5%, like I said, 1.5% codes for our genes, which codes for proteins. But there are 3.5% of human genome which is conserved, and that's very important. So now we can start coloring some more, that these are the important things apart from the coding regions, which codes for proteins. These are other regions that are important for survival of the species. And so what are these non-coding functional sequences? So we now know these regions as regulatory elements. So like enhancers, silencers, insulators, and locus control regions. So these are elements which are important for the right kind of genes to be expressed at the right time. So as you know, because our cells are all different, a liver cell is different from a skin cell and like that. So the set of genes that are expressed in each of these cells are actually different from each other in these tissues. There are, of course, some genes which are expressed in all kinds of tissues, but there are genes which are specifically for to be expressed in certain types of tissues. And these regulatory elements help in regulating which part of the chromosomes open up, and thus those genes can be expressed into proteins. There are also chromosomal functional elements. So these are like for packaging the chromosomes in a right way, because with packaging and because these chromosomes have to be passed on from one cell to the daughter cells, or from one organism to its offsprings. So these chromosomal elements are also important, and thus they are also conserved. So now we know that 3.5% of the genome is functional, yet non-coding, but and these constitutes gene regulatory elements and chromosomal functional elements. And there is, apart from that, there is also the epigenomic landscape. So the epigenome is considered like a like the jewels of the genome. So if you have if the genes are expressed, like if you turn on a fan or turn off a fan, like there's a master regulator switch, the epigenetic landscape is kind of like the regulator. So at certain point of time in the life of the organism, certain genes have to start producing genes, and certain genes have to be shut off. So the epigenetic landscape, they actually control that when a gene is being expressed and when a gene has to be stopped. So there was this project called the Encore Project, which started sometime in 2010, and this is a consortium. And the Encore Project was actually to understand what was there in the form of the epigenetic landscape. And after a lot of debate and all, what was finally we understood was that not just 5%, but it was actually 20% of the genome which is required, because these also have the carry the epigenetic signatures for the cells to function in the right way. Now we can think of the genome in 3 dimensions. So that was the sequence. That's what we so far we understood that this part of the genome. These are important for survival of the species and on. Now, what about variations? We are all not the same. White person is different from us, from a African American. We have different ethnicities, and even in a same ethnicity, if even though we are Indians, we are different, because we look different, we talk different, we have different kinds of characteristics, we have different diseases. So this is because of human genomic variations. So there are many variations in that 3 billion base pairs that we have. So I will come to what type of variations these are. At this point of time, please understand that there are variations, and between any and some of these variations are just there, sitting there, doing nothing at all. They're just there as variations. Some are actually protecting us. So some are helpful of these variations. So by variations, I mean that maybe some people have a thymine, T at a particular point in the chromosome, and others have a C there instead of a T. So these are variations. So these particular variations are called single nucleotide polymorphisms. And some of these are helpful or protective, and some of these are detrimental or cause disease. And between any two unrelated individuals, for example, between you and your friend who is actually not a family member but unrelated, the number of variations is about 3 to 5 million of 5 million of these single nuclear polymorphisms. Right now, let us call these variations because polymorphisms mean a certain thing to us. We will talk about that. So between mothers and fathers and sons and daughters, these variations are less. They are lesser than these 3 million because they are related, and also among brothers and sisters, because they are related, these variations are much less in number. But between any two unrelated individuals, is 3 to 5. So 5 million. So to map these variations, this was the first project which was launched. It was called the International HapMap Project. So this was mapping all the different kinds of variations that are in the human genome. And currently there are probably close to 1 million of these genomes which have been mapped for different ethnic groups. And we have a fair understanding of what the variations are and where they lie. There is also there were at the same time in 2010 the 1000 Genomes project was also launched. So this was not only mapping the variations. This was because there are and most of these variations are quite common variations. So the 1000 Genomes project was for understanding what were the rare variations were there. So the 1000 Genomes project entailed that they were going to sequence. So Human Genome Project was one sequence. So 1000 genomes had to be sequenced to understand the rare variants. This has been so successful that there are right now maybe 10,000 or 20,000 genomes which have already been sequenced, maybe much more. Maybe 100,000 genomes have already been, whole genomes have been sequenced throughout the world. Why are we doing all this? Because we need to understand the genomic basis of human diseases. So we understood where each sequence was in the genome. We understand that there are variations. Now, what does it mean for us? So in the genomic architecture of genetic diseases, there are two kinds of diseases. The first is called Mendelian, because they are passed on in the Mendelian fashion, like the pea plants. These are very rare diseases, like Huntington's disease, myotonic dystrophy. So these are rare diseases. These are monogenic in the sense that they are caused by defect in only a single gene, and these mutations are mostly in the coding regions of any genes. There are also non-Mendelian disorders, which are common disorders. For example, hypertension or diabetes, or fatty liver disease, or so these diseases are quite common in the population, and sometimes, for example, like diabetes is growing in the Indian population. So these are very common disorders. And these are complex in the sense that there are many genes which are responsible for these disorders. And there is also what we call the environmental effect. For example, diabetes can sometimes be controlled by living a healthy lifestyle. So there isn't. So that's your environmental component. So there is a genetic component and an environmental component to these Mendelian, non-Mendelian common complex disorders. And the mutations that we find are mostly in the non-coding regions for these type of disorders like diabetes and hypertension. If you go to this website, which is called Online Mendelian Inheritance in Man, It's the catalog of about 6,000 Mendelian disorders or traits that have been identified so far, most of which we already know the molecular basis, because we can sequencing has become so much easier. So there are a few of them which we don't know the molecular basis, maybe a couple of thousand, which we don't know the molecular basis, just for the simple fact that that has been cataloged at some time in the past, but there are no existing families at all which we can study to understand what was the mutational basis for the disease. And so coming to the variants, like I said before, so these are common variants, then there are rare variants. So if a variant is in more than 1% of in a community, then we call it a polymorphism, or in this case, it's a single nucleotide polymorphism. If it's rare like this, then we call it a rare variant. So a rare variant may be what people understand mostly as mutations. So what we now know is that rare variants or these mutations, they cause Mendelian disorders or single gene disorders, and common variants which are found in most people, they cause common diseases like diabetes, hypertension, etc. So how do you understand common variants? So before that, like even 20 years back, it was very difficult to actually understand the genetic cause of common variants, just because of the fact that we did not have the requisite tool to understand that. So now, because we have a certain technology called GWAS, or genome-wide association studies, we can study the variations in the human genome at one go. So if you take a GWAS chip, there are like 12, you can study the variants of 12 people in one go. And why do we need that? For example, if you look at these two genes, this is gene A and this is gene B, and a particular variant is in gene A among affected and the blue and purple, and affected and unaffected. And then in gene B, affected people mostly have the orange variant, and unaffected people mostly have the blue variant. So by that, we are, just by eyeballing, we can say that the gene B is probably what is responsible for disease, and gene A is not. So if you go and do this for the millions of polymorphisms that are in there, that will tell you which are genes that are associated with the disease and which genes are not. So that is the way we study common disease nowadays. And the first genome-wide association success story was published in Science in 2005. And since then, if you go to this NHGRI catalog, you will see all the different areas of the different chromosomes or the polymorphisms which have been associated with certain kinds of, whether it's a digestive disease or a cardiovascular disease. So these have been associated with those variants have been associated with those kind of complex diseases. So it's the first success, Ziva success story was in 2005, and since then it has been growing steadily through the years. This particular slide is a bit dated. Total number of publications just on GWAS has been growing exponentially since 2005. And now we know the genetic basis for a lot of these complex disorders. And what we also find interesting is that this set of mutation or variations, they vary from one particular ethnic group to the other. So the set of genes that variants that cause diabetes in Indians is not exactly the same as what causes diabetes among Caucasians or Japanese or Chinese. So these sets are different. There are some which are common, of course, but there are which are unique to different ethnic groups. So now that we understand variants and what causes different kinds of diseases and all, next step is to go for routine whole genome sequencing for all kinds of individuals. But then back in 2003, that the limiting thing was actually the cost of it. So the Human Genome Project took 13 years to do, and to complete, and the cost was $1 billion. So in 2003, so that was not feasible if you are talking about everyone on this planet. So in 2003, this is what they said in Nature, that technological leaps had to happen, and the ability to sequence DNA at costs that are lower by 4 to 5 orders of magnitude than the current cost, allowing a human genome to be sequenced for $1,000 or less. So this is what it is. The first Human Genome Project cost us $1 billion, and even in 2003, when the first Genome Project was completed, people were talking about the $1,000 genome. So sequencing the whole genome for just $1,000. So where are we? Yes, there have been many different technological bounds, and where are we in this figure? There is the next generation sequencing that has happened. So there are a lot of different kinds of technological improvements have been made. Chemistry has behind the work has improved. So we are somewhere now over here. So the current cost is about approximately $3,000. So we are $3,000 for sequencing whole genome, and the first genome sequencing took us 13 years. Nowadays we can sequence it in 3 to 4 days. So forget about years, we are talking about 3 to 4 days now, and forget about $100 billion, we are talking about $3,000 is the current cost of sequencing the whole genome. So now that we are almost there to the $1,000 genome sequencing, next comes routine analysis of the human genome sequences. So where are we in terms of routine analysis? So the current bottleneck in genomics can be explained by these two figures. So now we have a huge amount of data coming out, whole genomes being sequenced at within 3 to 4 days. But the computational ability to analyze the data has not caught up with the rate by which we generate the data. So it takes months for us to understand where what it means. The sequence is there, but it takes us months to understand what it means. There are problems like aligning where each sequence goes, and there are also other, because there are a lot of the Human Genome Project is very repetitive, so aligning them in the right order is one of the difficulties, and we are talking about terabytes and terabytes of data. So computational ability has not come up with our ability to generate data. So that's our computational bottleneck. So if you come to NIBMG or go to sequencing other sequencing facilities, you will see banks of these mainframe computers that are churning out not only data but doing all those, analyzing the data, compiling the data, and storing the data is also a huge problem. So that's the information bottleneck. So not only we have these whole genome sequences, we have to know where the variations are, and that means terabytes and terabytes of data, and storing the data itself is a problem. So nowadays there's cloud storing, but it is still a huge problem storing the data. So that's your information bottleneck. So you have a lot of information. What does it do? That's the issue. So in 2011, this is what Professor Harold Vermus said. He said physicians are still a long way from submitting their patients' full genomes for sequencing, not because the price is high, but because the data are difficult to interpret. So Professor Vermus actually inaugurated our Genome Hall a couple of years back. He was here at NIBMG, so he's a Nobel laureate. But things have improved quite a lot. So if I took you through those steps by which we not only understand the variants, we understand now what causes disorders, and the final step is because we are all different. What medicines will work for person A versus person D. So the field of personalized medicine, that is what we want to achieve to improve healthcare. And beyond, we are actually now in the fourth segment. So generating the data is not a problem. Interpreting the data is still a problem, but with better computational abilities, it will not be a problem in the near future. And then we can go for personalized medicine. And that will also entail new diagnostic procedures, new therapeutics, and new prevention strategies and whatnot. So that's the way we are going to realize our genomic medicine dream. So what about this? Maybe the rest of the Mendelian cause of the Mendelian disorders will be known. This will be the common complex disorders. The causes will be known for each different group and even for each different person. Right now there are two. I'd like to. So that was about realizing the genomic medicine gene. I'd like to talk about two different projects that are currently going on. One in which India is taking part. So the first is the International Cancer Genome Consortium. So the Human Genome Project was actually there were 8 countries which were involved. The International Cancer Genome Consortium is a 19-country team which are actually sequencing all the different kinds of cancers. And India is taking part in this. India is part of the International Cancer Genome Consortium, and we are sequencing oral cancer because that's the most common type of cancer that we find in India. Because we have habits like chewing tobacco and pan and those things. So oral cancer is much more prevalent in India, and that's why we are sequencing oral cancer genomes. And the sequencing work is going on at NIBMG, right here at NIBMG where I work. And there's a team in the coming months. You'll be hearing from Dr. Arindam Moitro, who is leading that team, so that work is going on. Cancer sequencing, all different kinds of oral cancers. The other project which I would like to talk about is the Human Microbiome Project. So if you compare the total number of microbiomes that are in our body, so these are tiny organisms that have a symbiotic relationship in a human body. The total number of such organisms outnumber us by 1 to 2. So maybe even more. So initially it was thought that there were 10 times the number of microbes in our body than human cells. So that would mean that only 10% of you consists of your cells. The rest is made up of other organisms. The thought is scary, but there it is. So the Human Microbiome Project was started to, and most of these microbiomes that are in the human body cannot be cultured outside of the human body. So we don't even know what is in there. But because of now we can sequence the DNA of these or the RNA of these organisms, we are coming close to understanding the different kinds of microbes that invade the human body, have a symbiotic relationship with the human body, and can also cause disease because the balance between the human cells and the microbes might change at any point of time, leading to different kinds of diseases. So we are trying to understand that. So there are different parts of the human body from where samples are being taken, and sequence the human microbiome project is going on to understand what type of bacteria or the other organisms are in there. So that's the project. So like I said, that what is the real reason of realizing the genomic medicine dream is because there are a lot of medicines that have already been out there that have been produced by pharmaceutical companies, but they're not out in the market just because they do not cater to a large number of individuals. So if you understand that who they can cater to, maybe these medicines will actually come to use for us. So that would be the final realization of the genomic medicine dream. I thank you now. It was a pleasure to talking to you, and I hope you all stay safe, and we will all get through these difficult times, and I wish you all the best. Thank you.
Thank you, Dr. Priyadarshi Basu, for his excellent presentation. So you must have written all questions in the chat box. Dr. Basu will look at it and answer your questions accordingly. Okay. Thank you.
Yeah, so I have noted down some of the questions which have been asked.
Okay, Alpana, so you can
yeah. So can you just narrate some of the questions, and then I'll
Yes, please. Yes, sure. So Sri Hari wanted to know something about the some status of the Human Genome Project, which I think you have briefly covered in your talk. The other question that he had was, what according to you is the most significant finding for the mankind out of Human Microbiome Project, Microbiome Genome Project?
You know what like I said before, most of the microbes that are inside the human body, we cannot identify what they are just because of the fact that they cannot be cultured outside of the human body. So if we didn't have the sequencing technologies that we have nowadays, it would have been impossible for us to even know how many there are. Now we are saying that the total number of microbes, they outnumber us by 2:1 or even more, but we had no clue before the start of the Human Microbiome Project of approximately how many microbes there are. The other thing which is very important, and I'm sure at one point of time, one of our faculty members who work on human microbiome will talk about this in greater detail. What we see is that, for example, the gut microbiome. So what's in the intestine? So the gut microbiome for a person who leads a certain kind of lifestyle, or is an urban person, or is probably vastly different from a street child, and so that also affects our immunity. Not only disease is caused by imbalance if we suddenly change our diet or suddenly do something that we are not used to. The microbiome imbalance will start, and that can lead to certain disorders. There is a like vast amount of papers now being published where the microbiome imbalance is being touted as one of the causes of these complex disorders. So along with the genetics and the environment, now we have the microbiome to worry about in diabetes, in fatty liver disease, all these common disorders. It seems that the microbiome plays a big role, and just because of this imbalance of the particular distribution of microbiomes that are supposed to be in the gut. I think that's a very important discovery, and I think in the future this discovery will actually change our way of thinking in the field of genomics.
Sure. Thank you, Dr. Basu. So Shivani has a question. She wants to know what is the difference between QTLs and GWAS.
So we humans have different kinds of traits. Let's forget about disease. Let's talk about traits and characters. So, for example, some of us have our earlobe, which is unattached from the from our face. The bottom is unattached from our face. Some have earlobes which are attached to a face. So it's either attached or it's unattached. So it's a dichotomous condition. So it's all or none. It's either there or it's not there. So that is not a quantitative measure. It's just there or not there. But for example, things like height or weight, so those are quantitative markers. So you cannot have something like a zero height. You have to have, even if you have the smallest person on earth, you still have a measurable height or a weight. So those are quantitative characters. So when we design GWAS, we can design it in such a way that it's a case and control design. So we have a bunch of individuals which are healthy, and we have a bunch of individuals who have diabetes. So if you're trying to find out the genetic cause of diabetes, so that's typically what a case control study is. This is actually how GWAS started. But now we also measure these quantitative traits, where, like I said, because for example, in the pea plant experiments that Mendel did, he was lucky because the height of pea plant is determined by a single gene. In humans, hundreds of genes actually are responsible for a person's final height. So for that, because it's a continuous trait, we consider those as quantitative trait, and this can also be done using GWAS. It's just a different kind of calculation that we do for them. Yes.
Yes, thank you.
Next.
Yes, so the next question is from Ashlesh. He wants to know that if a person is suffering from a genomic mutation and he takes genomic medicine, what effect will it have on its on that person's genome? So I think he needs a little bit of clarity on what genomic medicine.
I mean, genomic medicine does not mean that it's a specific kind of medicine. Even metformin can be a genomic medicine. What I mean by genomic medicine is because for example, let's go with metformin. Metformin is a drug which works beautifully for diabetics in European countries. Metformin doesn't work very well for us Indians because of the fact that we are genetically quite variant from Europeans. We differ between any two unrelated individuals. I already said that we differ, but we differ more if we are comparing an Indian and a Caucasian. So for example, a drug which was working beautifully for this population might not work very well for the other population. So because of the variant. So by genomic medicine, I mean that. But it's the same chemical type of. So it might not mean metformin. It might be some other drug which works well for us Indians. That's what I mean.
Yes, so these are the only questions. Okay,
Okay.
Another question from Shivani is, what is the key advantage of GWAS?
Looks like you are. So, see, Shivani, the thing is, even if you do a whole genome sequencing, that will cost you still $3,000 per thing. So, if you are trying to look at maybe 1,000 individuals and trying to figure out what are the mutations that in these individuals that cause a particular disease, you end up paying a huge amount of money. So it's $3,000 per individual is no joke. We are talking about 1,000 individuals. So instead of doing that, if you know that so the GWAS works like this. So because there are recombination hotspots in parts of the chromosome are actually segregated from one generation to the next completely intact. So it might be that if you look at 10 different variants in that particular segment, which are also called haplotypes, it will be same for all individuals in that particular community. So instead of looking at the entire sequence, why not just look at one of them? If it's a T or an A over there, then that means the flanking variations are also in that particular category. So it will be the same. So instead of sequencing the entire thing, you just look at one SNP. That bring down the cost to even 10 times less. So that's the reason why we still look at GWAS, and once we find that these are the regions after the GWAS, once we find that these are the regions which are associated with the disease, GWAS it does not point us to genes which are causal. All it points is to a particular regions of the genome which are associated with the disease. Yes, and after that, the next step would be to sequence only those few regions which are associated with the disease and not the entire genome. That will bring down the cost to a huge amount. That's the reason why we do GWAS and not go for sequencing from the beginning, because it's cost prohibitive, still cost prohibitive. Thank you. Thanks. Any other questions? Alpana?
Yes, so there's this one last question. I'm not 100% sure what Praveen wants to ask. He has written the energetic cost for alternative splicing is still unknown, and the same holds true for translation. How could this be answered? I'm assuming it is related to the energy.
Alpana, can you repeat the question?
The energetic cost of alternative splicing is still unknown, and for alternative splicing and also for translation is still unknown. How could that be answered?
There are two ways of which we do that. One is we have all these scanners. Like I said, we use these kind of scanners for doing GWAS studies. There are chips also, microarray chips also, which are for transcriptomes. So what we can do is, so basically these transcriptome chips, they tell you what kind of genes are being expressed in a particular tissue and to what level. So we can even compare these two kinds between normal individuals and normal tissues and diseased tissues to know what kind of genes are expressed at a higher level or what kind of genes are expressed at a lower level, doing similar kind of thing. So we can use the microarray chip for looking at the whole transcriptome. That's one of the ways. The other way is to go for something called RNA-seq. So this is basically sequencing the RNAs that are being expressed in particular tissues. So in this case, you not only understand how much of those genes are being expressed, you also can find out if there are mutations that are in the RNA itself. So by those two are the approaches by which you can look at all these different transcripts of genes. So there are 22,000 genes, but there are many more transcripts, like I said. So maybe 100,000 transcripts at least, but we can look at them at the same time using either a transcriptomic chip, or we can go for an RNA-seq, which is of course again very quite expensive. So it's like the difference between GWAS and whole genome sequencing. So these are the two approaches by which we do it currently.
Thank you, Dr. Basu. These were the only questions.
All right. Thank you, everyone. Stay safe, and thank you very much.